Dr. Judy Mikovits, you have been a research scientist and so much more for so many decades. And you've been in what I call the belly of the beast in terms of the institutions of science in the United States. In my work as a kind of investigative journalist reporter on that, I saw so much corruption from the outside and recognized it. But you were on the inside of it, witnessing it firsthand, seeing it, the corruption of science. And this isn't an accusation that all science is bad in any stretch of the imagination, because it's an amazing endeavor to do. But how to navigate that world? How did you see what you saw, survive it, and then come out so that we can look at both the microcosm and the macrocosm and make sense of it all?
Right. Yeah. And it's not so much the corruption in science, it's the corruption of medicine, because medicine isn't science. And science, the translation of science into the practice of medicine is what's been corrupted. And it's been corrupted by, as we know, big money and big tech at every level. So technologies always precede advancements in medicine.
Right.
When we're able to see things at a molecular level. So when I started college in 1976 at the University of Virginia, their arts and sciences program didn't even have a bachelor's degree in a Bachelor of Science degree in biochemistry. So I had to get a Bachelor of Arts degree in chemistry with a specialization in biology, and about all I could study were plants. We had no genetics. We had one known genetic disease, phenylketonuria, biochemistry genetic disease. And so that phenylketonuria is a metabolism disease where because you can't metabolize phenylalanine as quickly at the amino acid branch chain, you get a retardation. You build up a problem in the brain because you have too many ketones in the urine. So all we would do is a heel stick for kids.
Right, the PKU test, they would call it.
Correct.
Yeah.
And you just check, and then you take all the phenylalanine out of the diet until they mature, until the brain finishes maturing and the immune system.
So that's a great example of learning a biochemical pathway that is corrupted and applying it medicinally, if you will, in a procedure that could prevent something horrible from happening. And
Correct.
So a proper application of a scientific discovery.
Correct. That's science and translational medicine. So I went from the University of Virginia, where my specialty was plants, because all we had was plants. So purify medicines from plants and soil and natural products. So I was a natural products chemist in my first job at the National Cancer Institute as a entry-level, low-level technician, the lowest level technician you could be there in 1980. At Fort Detrick at the National Cancer Institute. I answered an ad in the Washington Post to purify interferon. And interferon was on the cover of Time magazine, March 31st, 1980, that year I graduated, a month or so before, as the big if. It interferes. It's a protein that interferes with viruses infecting cells. It's an interferon. It's in the innate level. It stops poisons from getting into the cell, just like shutting down. I think of Katie bar the doors, just everything shuts down. You shut down the fort, you pull up the moat, and nothing comes into those cells, primarily the stem cells. So I'm just fermentation chemistry, natural products that requires alcohol, glycolysis, sugar being broken down.
I remember
in fermentation and you burn off the alcohol and you end up with the nutrients and these are our biochemical pathways that they could only teach the pathways and you could only do what nature did. So you had to understand cows, pigs, biology at every level and how those immune systems differ.
I'm flooded with the memories of 1980 as well. I was still in high school at that point, remembering that Time magazine article because I was a science kid. Yeah. I loved diving deep in. I'm like, oh, interferon, this could be the greatest discovery. It could be a new thing to prevent disease, all that. And there was a lot of speculation about it. And then as it was put into play in terms of a synthesized version, perhaps, they found numerous side effects. It wasn't doing as well as they thought it would. And it just was like, well, that wonderful discovery may not be so wonderful.
And absolutely, you just discovered the corruption. And what the corruption was we purified, I made cell lines, we purified bovine, human, pig, other kinds of interferons. We purified them in fermentation chemistry. Literally, I collected cow snot. And so you would blow a virus up the cow's nose, various families of viruses, just think things like measles and influenza, flu, those viruses that Professor Dolores Cahill shows you, they're not enveloped. They don't float through the air. They're killed by sunlight. And so they change very rapidly. They never come close to infecting you, even if they land on your skin and hit your natural oils and viral. So what we discovered is that according to God, and we knew this, that God gave us everything for food and for our well-being. Well, cow interferon, every lower interferon from every animal that I purified, just cut off in 100,000 molecular weight filter. So you just cut off everything that size, and all of those interferons, not any specific one, worked in concert. So the cow, just in saline, which would cost pennies, and I'll tell you the product right now and where the corruption comes in, and we can see it, but that worked on all humans. And the recombinant, when we took the gene and we expressed the gene, which we were only able to do crudely in the mid-'80s. So the cow interferon, the bovine interferon that you couldn't patent in 1982 because of the Bayh-Dole Act in 1980, which every one of us in that lab in the biological response modifiers program at Fort Detrick were absolutely unaware of that law. We did not know of that. So we could not understand why nobody would use the pennies on the dollar bovine interferon that is a nasal spray and a throat spray. You don't inject it. But they had to inject it because without post-translational modifications and post-transcriptional regulations, that would be after the mRNA. Its DNA is made into mRNA. It is exported. The message is exported to the cytoplasm, away from the nucleus of the cell. It's then the messages, the microRNAs, the small inhibitory RNAs, the long-chain non-coding RNAs, through the endoplasmic reticulum, through the highways. They modify, they put sugars on, they do other things, and then they export from the cell the enzymes, orchestrated by God, according to the environment. You're a cow, you're not a human. But a cow interferon will work on a human. Human interferon purified from me will not work on a cow. That cow will stay sick, continue making more interferons, and even make more. So we would do the natural experiment, according to God. But the government couldn't patent it. And the Bayh-Dole Act allowed for these public-private partnerships. So people would buy our discoveries and change a nucleotide or two, or even as they saw them coming through the publication process, they would corrupt the data, change a base pair or two to say, I discovered it. No, you discovered it. And no, lymphadenopathy virus was never human immune deficiency virus. So therefore, you have the test that tells you which variant, and you can follow that test for the treatment, and everybody makes a ton of money and nobody gets well. Yeah. And that's now starting with that interferon, which is called Paximune, P-A-X-I-M-U-N-E. And I hope you will get an opportunity to interview either Dr. Ali Muhammad, because there he and Dr. Joe Cummins, who can't talk to you right now due to Parkinson's
Sure.
disease, can tell you, and this is Kent Heckenlively's book, Inoculated. So once you see that it started right there corrupting God's discovery, Time magazine was good then.
Yes.
It said the big if, and it was a very well written article.
I remember
And yeah, you and I remember it like it was, but
Yeah.
people don't remember now. And all they think of is injected Interferon or the various recombinants. And the higher the unit, it shuts down those natural sensor pathways that are feeding back from nature. So you have to use more and more to shut off the fire, and you can't. And you become desensitized, and you start attacking yourself really in your production capabilities at the level of the stem cell.
So if I'm to understand or correctly communicate this, you had a form that would work in more of its natural state, so to speak, that when they realized they couldn't profit from it, that's where we get the corruption of the medicine back end of it. You had a scientific discovery, amazing. And they alter it. And this is where we get into what synthetic type chemistry.
Correct.
It's like, and so it changes it from its natural state where it was workable to something that could be sold for very high profit margin and yet wouldn't work, but fed an industry that survives on sickness, not an actual correction of what's gone wrong. And then since that time, we've seen the regulatory agencies completely captured, and the scientists who are amazing scientists, if they were proposing subject investigations on something that would go outside of perhaps that what we call maybe a pharmaceutical box, but the idea that we could synthesize, patent, and bring to market something, that the funding would dry up immediately, even if you were a solid scientist of great repute and renown.
Absolutely. So, and that required the publications. So what next had to happen? Okay, so you've got the Bayh-Dole Act that none of us are aware of, hardworking scientists, the biological response modifiers programs, MDs, PhDs, PharmDs, nurses, doctors, me, a lowly technician. I did the adoptive transfer. I isolated from saliva. I just took the saliva and manufactured there in those biological cell line systems, fermented LAV from the saliva of the French Canadian, pretty much patient zero over in France. And so I got, and I'm collecting cow snot and I'm in the face, no mask, of the guy that supposedly has AIDS in this virus. But knowing your immune system and knowing the protection you have in your mouth microbiome, this is what we're starting to really appreciate it. So at the literature stage, the reviewers, I call them the gatekeepers of science, the reviewers of the publications, as your so-called peer review. Well, if you've discovered something, you have no peer. So you've only a competitor who's jealous, as we hear really about Cardio Miracle, as we hear this is that story. Oh, you're not a scientist, John. I call it the download of God. And why is my experience? So you're here. And so in the early 90s, not only do you have to have a peer-reviewed publication, but there's an impact factor attached to the journals. So now the gatekeepers of science, the journal science, the journal's nature, they decide what it's worth, how much is paid to get it into the profit, into the those will determine not only your funding, but those all important Nobel Prizes.
Right.
And I'd love to tell the story of reverse transcriptase, Howard Temin, the Nobel Prize for reverse transcriptase, which is the key enzyme, God-given for all mammals, that reverse transcribes, writes backwards your RNA into DNA, and then inserts it back into the genome in order to correct errors. God didn't make oncogenes, cancer-causing genes. Let's just clear that up. No such thing as breast cancer run or a prostate-specific antigen. All our genes are written and transcribed according to our environment, and it goes back 4 generations where your immunity and your brain are inextricably linked, and you can sense those poisons according to your environment as long as you don't desensitize them. So David Baltimore So he's now. I'll just mention the book Emerging Viruses, Lenny Horowitz,
Yes.
1960 or so, telling the stories back to the 60s, because it's important, because I just mentioned that I didn't get to the National Cancer Institute till 1980. So I have to see these guys are gods, little g. They're gods, little g. They've assigned it themselves. They're the peer reviewers. The buck stops here. They're the editors of the highest peer-reviewed journals. So they come in here, and David Baltimore, Science. So Howard Timmons at Wisconsin, the discoverer of reverse transcriptase, submits it to the journal Science. And of course, David Baltimore sees it come through as the senior editor and says, there's a Nobel Prize. So they write reviews to buy time and say, no, I'm 95% convinced, something like this, because I know the reviews I used to get. One was, I didn't believe your other paper either. Sorry, it's not religion. So the other was, I'm 95% convinced. Show me an immune response. So they see it, but they don't yet understand it. And then then while you're out trying to show them the other piece of data, which can sometimes take months because you have no technology. And these papers determine your career, your lifestyle, and most importantly to all the men who care, Nobel Prizes, because women never get them anyway. Rosalind Franklin discovered the crystal structure of DNA, and Watson and Crick just, beat up the woman, woman is nigger of the world, John Lennon. Anyway, just saying, that's how it works in science too. It's no worse. But so David Baltimore sees the paper.
Yeah.
He assigns a project to change a few key base pairs. And see if we can modify this and write a pretend paper, a companion paper, and get it back in from one of his unknowing postdocs or senior scientists. You see, these guys, they see what comes through. And you see this in our book, Ending Plague, so that I'm not telling stories, because Frank Rossetti was there. They all sit in lab meetings and say, who wants to reject so-and-so today? Or who wants to take the ideas from this grant, which we rejected the grant proposal at a university and professors won't exist? Who wants to take this idea and make it their own so that we can patent it? Patents, Bayh-Dole Act. And then we can patent it, we get credit, we share the Nobel Prize, and you just off the natural discovery. That's exactly what David Baltimore did with Howard Timmons and that paper. If you take David Baltimore's science paper from I guess it was the 70s, early 70s, and try to do the experiments, replicate what he did, you can't do it. You can't do it.
So what we're talking about is a closed loop of corruption.
Correct.
The people that rose up to the top had to play that game, or else they would be drummed out.
Correct.
And therefore you had a fiefdom established, and you have the character known as Anthony Fauci, somewhat doling out, if you will, that budget. Where are we going to send this money? And it's going to be sent only to those who are playing our game.
Correct.
And therefore you have how many lost decades or perhaps centuries of advancement in that time frame, because the technology was emerging to discover the things you were witnessing and discovering.
But the key is the technology never worked. And so the key and the fun of all it is cardio miracle. That's why, and that those of us who never stopped. So I consulted in 2005. So I got married October 7th of 2000. I was just so done with what I saw. I rose from that entry-level technician to the head of the lab of antiviral drug mechanisms, where we use those very technology. Because you see, they were playing them all along. And that lab of antiviral drug mechanisms in 1999, the entire lab quit. They were led by Bill Rice, Jim Turpin, and others, lab of antiviral drug mechanisms. And the whole lab quit. Why? They were making all the discoveries, and the discoveries were being shipped to places like San Diego. Oh, La Jolla. People were given credit for patents where they were functional forgeries, as our friend David Martin showed us in Plandemic Indoctrination. So you take what nature did and you change it and you call it proprietary.
This is
Oops.
Fundamental violations, thou shalt
Correct.
not steal. Right?
Correct.
These are people that have, if we talk morality, they're amoral, I
Correct.
think, in many ways. I perceive that they may be atheistic in their worldview because they don't feel right and wrong matters. It's only what they can get away with
Right.
and can do. And so we see, and that's why I bring up the advancements in science, if you will, and technology aside, again, genuine things that were actually coming to pass that were remarkable and marvelous, that were curtailed because of the economic implications.
But where God comes in is we never lost those data. Where God comes in is technologies, Steve, only show us how more right people like John Hewlett were. And then, again, I'm just going to keep calling it a download from God and the good people, the genius, Genyous, Chinese guys with a spelling problem. Well, they had the best marine biologists. They had British Columbia. They had people from Big Pharma. Jerry Norskog was J&J 30 years. He made fentanyl. And in 2000, he said, people are dying. Jim Dow, the head of a Chinese, and they were in these Stanford and made things like electron micrographs. They were pioneers of their time, and they realized there's got to be a better way. So when the Holy Spirit touched them and said, no, this isn't, we can heal people. And at our root, we go in to do good. Yeah, glory and money is great, but we do it to do good. And there, Jim Dow set out in 2005 to have, maybe even 2000, but his first patent was 2005 on natural products. Let's use what botanical adjuvants. Let's take little bits of everything from plants and protect the pathways of pathogenesis, of causing generic genesis, the genesis of disease, and bring them back to pathways of healing designed by God, and just use your knowledge of God's tool. So the funny in all of this is the technology keeps proving us right. We didn't know why bovine interferon, and the Satanists just said, the satanic cult of scientism. It's not it's an ideology, because it's certainly in their religion, and if you don't obey, it's a pretty deadly game. So that knowledge, we knew bovine didn't work. We were trained in the Bible. Nope, everything given by food, everything given for good. But you also have to treat everything according to God. You also are responsible for that soil, for that land, for that water. So we knew it. And then the technology proved it. And it's like, bam, bingo. So it's actually quite a lot of fun because we can go back. And right now I'm just, enjoying messing with all their heads, because it's like, find me another. So, and they see the papers are 90% right. So where the crime comes in is you couldn't discern that about David Baltimore then. I suspected it then on the political end, because you can see it go through there and it's like, you didn't do that work. Howard Temin did that work. And my friends were in the lab. We were a generation younger. That's 15 years, because you go through that much before you become that level of scientist. So you're just saying, and it's not right. And you just keep it there, and you lean not on your own understanding, but you wait on God to tell you when to use that knowledge. And that's the key. So when the good people and when people are a little more accepting, it's not Fauci. Fauci was just one of many mafia dons. Yes. And the Cancer Institute was so upset that NIAID stole all the Cancer Institute patents with Bayh-Dole. Purinergic modulation. That was Jeffrey Bernstock in 1972. That's purines and pyrimidines, sense of taste, sense of smell, ATP as energy, as metabolites. That's God-given, the base pairs of DNA of all of life. And that's a big deal. So when you understand basic, and then the technologies allowed molecular biology. So in 1992, I get my PhD in molecular biology and biochemistry. My other degree is in chemistry. And so that was 92. It took that long to get the technology, and everything I learned working with God, technology, and then you could see it. You say, hey, doesn't matter which virus they create. Keep it silent, keep the immune system, keep the cations, ions. It's just minerals. It's minerals according to the genetic code. Whose genetic code? Each individual is sovereign. And that's what we could do now with these technologies, with Cardiomiracle as a foundation.
Well, that's the clinical experience I had, especially through the 90s and early 2000s, working with severely immunocompromised patients, AIDS, if you will. And I ignored status of a test because it was irrelevant ultimately to, are you functioning?
Right.
Do you have the flow of life through you? And what are you missing? And what's there that should be removed? And I say, these people are living. They're not only surviving, but they're thriving. And so it wasn't a kind of a hunt for, the minutia, if you will, that you can find scientifically. And I see, wow, that bigger picture. How do we come back out to that? And this has been part of that discussion of how science is so awesome, because you can go down into that microcosm and see that one thing impact that one thing. But unless you come back out and see what else is related to it, you can end up creating more problems than you thought you solved because you saw that one pathway.
You never want to leave the forest for the trees.
Exactly.
And so how we never left it were people like Joe Oppenheim, Candace Pert, Luc Montagnier, Frank Rossetti, the people, whether they were God-fearing, outwardly Christian, as you were saying earlier, or just people that knew right and wrong and honored integrity. Yeah. So the problem isn't, but we stayed in that little lab and they left us alone. So, they considered me as either insane or, let's say, a drunk. I was pretty angry. So It's all in the book, Ending Plague. Yeah. You can hear all the fun we had. But, so when our friends, Bill Rice, Dr. Jim Turpin, closed the door, locked the door of the LADM in 1999, or the end of 1998, I was in a little bit of trouble for threatening a guy in a wheelchair as he was attempting to take things from the laboratory and twist. What was more important was take the knowledge, take the papers, change the meaning. Because you see, most of the papers are right. How do you tell a counterfeit? Hold it up to the light. So the words are very close that most people wouldn't recognize how the journal editors are changing the papers, the wording, the meaning. And this is what our friend Dr. Brian Artist can do. Start reading those papers, but we have to teach them to read differently. Notice that between the abstract and the title, the words, those are the editors. That's the commercial. Get down into the material and methods. And you can't mess with the scientists because they don't know what they're doing. And so it's because the Faucis and the Baltimores, they're not down there doing the experiments. They don't have that gift and that blessing. They have no idea. They never did an experiment. Or if they did, it was before we had these technologies. Sure. And so that's where, those of us must become emeritus, if you will, who are up there, because I can't do it anymore at the level quickly enough, but I can think it, and I need those kids to translate. We need to bring in with the love the younger generations. Let's get those 15-year younger and then show them how much fun it is to heal people, how quickly you can do it. And with Cardio Miracle, it was simply, I heard John, and I'll listen to everybody because I'm always looking for Anustat, and I've thought like Jim Dow of Genius, botanical adjuvants. You don't manufacture one at a time. Mix them in, know your families, plant families, know your soil, know your water, and put them back in according to God and nature with knowledge of the innate immune system of each living organ system. And that's where all genetically modified organisms are in fact vaccines, which would mean they're poisons. And so we make, and so vaccination is not an immunization. And we have to work on our words. We don't mix the two. If you want to use the end of a needle of poison, then the vaccination of Interferon or manufactured interferon would not replace the immunization of Paximune. Bovine interferon, and the government, literally ran Immunity out of business back in the 80s, as it would have been a major cure-all for HIV/AIDS, as was peptide T, the beautiful Dr. Candace Pert, and all. Now, all of these late grade, I'm the only one left, which gives you a little bit of only pause that I've got to learn to translate this faster because we don't have the luxury of time. But when in 2022, John, and a paper was published, an independent analysis of function of CardioMiracle. I call it an AI paper.
Yes.
So you looked at the nature according to how it was put in, and now we have the technology to do it. I knew it from just looking at it, and I knew it from what we'd done at Genius that we have a patent for back to 2011. Work they started in 2000, as I mentioned. So botanical adjuvants and what we call a smart platform. So all I have to do now is mix and match on a foundation of CardioMiracle. Why? Because that functional paper said you hit the three main pathways that Dr. Rissetti's entire life's work. He got the 1992 Distinguished Service Award for TGF beta, transforming growth factor beta, as the general of the hematopoietic, the blood stem cell. So if you can get out of the flame, if you're not inflamed, if you have balance, it's not an on or off switch. And it works through what they call a non-canonical pathway. Oh, that would be the endocannabinoid system, your God-given dimmer switch, lipid signaling system, CB2, the inflammatory from cannabis, what they won't let us have, working right along with your innate immune system, your type 1 interferons. Keep the flames low. Communicate with the various stem cell populations and tell them what to do and when to do it. So CardioMiracle hit the three key pathways. And I remember this paper and the publication showing all the lines, and all I'm looking at are the sensors. And right in the middle is TGF beta, and it's a yellow light. Can't be a red light. Can't be a green light, has to be a yellow light. When you hit that major pathway, is the inflammation enough to heal? Or do we need to stay here a little long because we might burn out other tissue? There's no such thing as autoimmunity. You don't attack yourself. So when it looks close enough to non-self, and when you don't have too many of those synthetics, then you can escape that and it can become a healing. So in the prostate cancer patent for using Casodex, which is bicalutamide, and all you have to do is look up the chemical structure. It's a pill for prostate cancer. Well, it's off patent. Synthetic used it at too high a dose, and all you needed to do was build an anestat, not a thermostat, not up and down, balance, homeostasis, the key to life. Just keep it at a yellow light. And here you are. And then enos, not nitric oxide, endothelial nitric oxide synthetase. There are three nitric oxide synthetases known. Neuronal, number one, your brain. That one better be working or nothing else will. You'll be a chicken with a head cut off. And you'll run around for a little while, but it won't orchestrate. So number two, inos, inflammatory. Number two, in the middle of the brain and the blood stem cell is the flame. Turn that flame down. Get rid of the poisons. Anything that's robbing oxygen from the red blood cell is a parasite. You make 10 to the 9th blood cells every day. Which one's red or white? And how do you determine where to put the fire out and which stem cell to drive according to the plant? It's easy. It's not rocket science. It's simple because Frank Rissetti and I did that from plants from the original. Frank Rissetti also in that 1992 Distinguished Service Award for the discovery of interleukin-2, which was called T-cell growth factor. You can't grow T cells in the laboratory and understand them until you can. So understanding those interleukins, communicators between two white blood cells. So they named them all 1 through 30, but we still remember when they were macrophage activating factor and macrophage colony stimulating factor. We know that language because I had to sit there for 22 or 30 years with Frank and figure it out since 1983. But I had my expertise and he was willing to listen. And even if I screamed at the top of my lungs, we would fight about what the meaning was. The meaning. And he's like, prove it. And so, and I'd always throw it up there. So with the cannabinoid receptor controlling TGF-beta1 on the blood stem cell, this was 2015. And Frank's writing a review with one of our lovely colleagues, Stephen Bartlemas, and they're frustrated because nobody understands TGF-beta. But more importantly, they're changing the meaning of TGF-beta science to forget those 1990s. So they're skewing it all to make young scientists believe something that's not true. That is what all those discoveries proved was not true. But then you'd only know the canonical, the cult of scientism. Yeah. You wouldn't know the non-canonical. Because he's like, Judy, I don't understand it. TGF-beta is signaling. And there is no measurable receptor, TGF-beta1 receptor on this stem cell. And I'm like, well, Frank, it's one of those squiggly little things that crosses the membrane 6 or 7 times. Maybe it's a cannabinoid. And he screamed at me, because we're obviously, he's like, there's no evidence for that. And my husband was sitting in the room just laughing as we're doing terpenes, our cannabinoids. All cannabinoids are terpenes, but not all terpenes are cannabinoids. So we can mix other plants and tame the THC, the psychoactive, and allow you just to tolerate the people that are irritating you. So we do this regularly. And so David's laughing. And so Frank leaves. Oh, that's ridiculous. You were here to help me. And he goes up the street to his house. The next morning we go to coffee at 6:00 in the morning. And he says, I hate you, Mikovits. And I said, you found it, didn't you? Which was the publication that proved TGF-beta on the hematopoietic stem cell went through the endocannabinoid sensor CB2. And so that cannabinoid sensor too, and it would feed back as it broke down ATP to ADP to AMP, and the adenosine outside the cell says, we're all good, guys. And this is the work of Bob Navio. So once you understand molecular biology along with biochemistry of all of life, then you got it. And there's nothing more to do. ENOS and the third pathway of Cardia Miracle in that functional paper was calveoli-mediated endocytosis. How do cells take up a virus as a single molecule, not as a clump, not as an inflammasome, not as a, what do they call those, whatever the bilayers or things that we don't clear with parasites. No, that's building snot around the bad cells so that you can excrete it. Yes. So this is what, once you change the language, so I can see language both ways. And that's, I'm probably the only one who can, or is at least gracious enough to try. I don't know what the word is according to God, but it takes a lot to read these papers. They hurt. They hurt. But when you see how they did it, if you can just humble yourself and look for the needle in the haystack. And I just want to say Brian Artis, because that's what he humbled himself to be willing to do. Yes. Look for it. And then allow me to do what Frank Rossetti did to me. Let mentor him by saying, okay, son, now look at this one, because you missed something here. This word, it's not a one size fits all. Your God made every nicotinic acid receptor for every individual system. So that's the fun of all of this.
Well, coming back out to that bigger picture, recognize some of the thematic elements of bringing our friend John Hewlett into this is that he's not a formally trained scientist of any kind. And yet, what a breakthrough. What do we see in history? It's not always coming from within institutions with people that actually have the degrees to do the science. Isn't that fascinating? Because it's a God connection, right, as we always bring up. And that's the beauty of this, because the people that knew about cannabinoids when they were being denigrated, they knew because they knew, they experienced it. And yet it took what?
1994, we sequenced the genome and we recognized the endocannabinoid system. Yeah. So those technologies used to recombinant interferon turned evil allowed us to see this. And actually we were those people, and we were with international people, and a lot of the cannabinoid research went to Israel. Again, shipping the research for natural healing over there and denying it here. And this is the corruption of the Faucis and the FDAs and the CDCs. They're experimenting on us and using our technologies. That's why Bill Rice left. So he went to a company, made a bunch of money, happened to be that San Diego company that cured Ebola with a little monoclonal antibody that came right out of the lab of antiviral drug. So we had it. And we saved all the samples. See, that's the key of pandemic. When I got married and moved, and plague of corruption, and moved across the country, I was able to take all of the work of the last 40 years with me to Epigenics Pharmaceuticals. And there, 5 years later, we met and collaborated with Genius Biomed right in Santa Barbara. So because, that's where we have the most beautiful plants next to Brazil. And so when you work together, and those people, we never appeared, nobody knew who we were. Yeah. So we weren't bad. We were just a little company that changed everything. So 911 destroyed both of those companies. Well, it destroyed Epigenics Pharmaceuticals because nobody cared about curing cancer. And we had a lab built in Carlsbad. We had houses and things, and we basically lost everything. But then we still knew these. We met these guys. God sent us these angels, Jim Dow and Jerry Norskog, and we just worked up and down the coast. And when I was jailed in 2011, all I couldn't say was XMRV. So I simply changed the name and just went with the pathways and knew we could heal it. No matter what chimera they make, we got you. Because all you have to do is never inject it again.
Well, I pointed out so many times over the years on my show that if you actually can help people overcome what we call disease, you can name it cancer, you can name it AIDS, you can name it any number of things. These federal agencies that are captured would find you like they have a time machine yesterday and stop you, right? Yet, this is the funny thing. It's not so funny, but it took them like 10 years to find who they claimed was behind 9/11. Right. Osama bin Laden in a cave on dialysis in Pakistan. I mean, just so many absurd stories that we believed that maybe now we're breaking through. Mm-hmm. And I think I come back to the spirit, because all of our worship of the mind has led to deception. As much as it's a tool like a computer, when we worship it, it becomes something that is amoral once again. Mm-hmm. And we see the corruption of the application of good scientists doing good work. Mm-hmm. It's stolen from them. These people have abandoned the spirit. Mm-hmm. And so we come back to a person like John Hewlett, Cardio Miracle, like the spirit spoke to him, gave him something that the scientists are going, how did you figure this out? Mm-hmm. And so what a beautiful story this is to tell. And of course, your journey is such an important story to tell. I'm just so grateful every time I get to connect with you and get a little bit more of it to come out, because it's the history of significant parts of the latter 20th century into the 21st, the corruption of what we're looking to remedy and are remedying one by one as somebody wakes up from the heart and recognizes it's the spirit that guides us to all of that brilliance.
Right, the heart, the soul, really, because the soul directs the mind. So it's easy. I took one look at that, having done this for 40 years. I took one look at that 2022 paper, and I said, that's it. And I couldn't call John fast enough. It's like, hey, I've been making these things forever, and there's no way I put that in that combination. So I hit the 3 key things of all things COVID. That's all we needed. It's like, well, so what do we do now? Because now all I have to do is put the rest. Put the rishis, put the terpenes, put the mushrooms, put the ivermectin, put the purinergic modulators that we already made. Because you see, this is the key. Those 1 drugs at a time, highly purified, can be the sniper. So you're protected with your infantries, with your soldiers, with your Air Force. Now you can go in and get that cancer stem cell. And we can do it now and absolutely cure everything right now. And that's the key. As long as everybody, no more injections, not ever again until you're, yes, sometimes you need an ER of an infusion of Meyer cocktail, but no, just saline, hydroxychloroquine, Lulaprevir, those drugs, there were no FDAs, ivermectin in, 1950s serum. And, we can make macrocyclic lactones, folks. Don't ask for serum. And they poison you when they inject it. We'll give it to you in a form.
Can I ask you a question that's like one of the most common questions I get when I bring up the subject of blood and coagulation issues and all of the various rat poisons they put in the bodies to stop, keep the blood thin, right? They're worried about very quickly on the coagulation concerns, the blood clotting, and all of the poisons that are put in to people because of that concern. Many people in what we call the natural realm have adopted this thing called nattokinase. And you've spoken, hey, this is a poison.
Correct.
I think we need some clarity because there's a lot of confusion out there. If you can do a brief version.
Sure. It's because it's not a kinase. It's fermented soybeans. The paper is 1980. So when you ferment, I'm a fermentation chemist starting in 1980. So when you ferment soybeans, you will get a white paste on them, and they're disgusting. They're a delicacy in many parts of the world because of extended life. And they affect a class of proteins called sirtuins and modifiers of DNA of message at the epigenetic level. I'll leave it there. But so that's what fermented soybeans is. Well, what nattokinase is it was made into a drug where we identified the exact microbe that metabolized fermented in the soybean, and that microbe would make those proteins stimulate. But they're disgusting. They become a poison too much. You can't do it if you go try to eat a lot of those. So, but when you stabilize that and you make it a drug, so it's only going to kill that one microbe, and all the other things that taste disgusting and are poisons you shouldn't be eating. And then you put it in a pill. So immediately number one, it's a serine protease. It'll cut after serines, every protein with a serine molecule. The key protein in our body we can't make in response, glutathione. Serine, got to break it down at the end of the dimethylglycine methylation pathways.
So there's a corruption of that important pathway.
So now you're going to break that. So you won't be able to make enough glutathione. You're just spinning out of control.
Which is a huge risk factor for a worsening scenario. So
correct.
We'll talk about that some more at another time.
Yeah.
We're talking about water, I think.
Well, I call Cardio Miracle living mineral water.
Yes.
And the reason I call it living mineral water is because the minerals aren't synthetic. The minerals are our lakes, our ground, our nature is all poisoned by the glyphosates, the heavy metals, what's raining down. So we the synthetic minerals. No, and I recently read the book by Dr. Richard Orley, our friend, Minerals for the Genetic Code. And I'm like, that's it. But I read the book after I decided it was living mineral waters, because all it is plants, freeze-dried, lyophilized, freeze-dried. So you get the oil, and at the nano level, at the electron micrograph level, you still maintain shape. So you can see things in nature, buckyballs, that's like C60, we call it. So in nature, it's the shape of water. Everything at your innate immune system is patterns and shape. And so all you're looking at is color, pattern, and shape between a nanometer of gold. So the reason I call it living water, and the reason I say it's a download from God, I looked at those ingredients. I didn't look at what the FDA makes you put on there, because I know that's BS. And I'm looking at function, and I can see the function in the three key pathways. The pine bark, why did you put those terpenes in that amount? 900 grams of this, a bit of that. So it's just plants. And then when they're all dissolved, it's so finely made into such a fine, and this is again technological advancements, that when you breathe the dust, you start sneezing. And one person said that to me. She said, I can't take that. Every time I open the bottle, I sneeze. I'm like, exactly. You're opening up your brain. Breathe it in slowly. I'm like, did you try putting something up and then pouring it in the bottle? And, okay then, and it worked perfectly, because you see, we don't get the living minerals according to God. We call it dry eye. We call it bone on bone. No, it's synovial fluid. There's no such thing as bone on bone. My favorite orthopedic surgeons and I always say that. No, you just didn't make the liquid. You didn't make the plasma. You didn't get the right nutrients in everything in our body that is not blood, is water. So we're fat because our fat cells are holding everything, because we don't get minerals. The key fat soluble minerals are A, D, K, and E. Where do you get them? Bee pollen. Dirt, leaves, trees that are pollinated, that are made from plants, cleanest plants you can get. Clean it up with the technologies. You can't go out and buy all these things in juice. I'm sorry. That's not how it works. And that's what we did. we lyophilize these things, freeze dry them. They're stable like forever because that's how God made it. In freeze-thaw cycles of our world, we preserved all of life all the time. So I call it living water because that's all you need to drink. Moving forward, no matter the poison, then you can carefully look at your own environment and add the food because you do need nutrients, but they're not calories. You don't count calories, you count chemicals.
Yes.
You know, count chemicals because at the heart of all energy is zinc, magnesium, calcium, ions, according to plants.
Right.
You don't take them, you eat, you drink them and you eat them, but our soil, our water, it just doesn't hold.
I loved it when you would yell to John at these conferences, "It's food, John, it's food," because it is the sustenance through the living waters, if you will. Yeah. And that's, you know, a cornerstone of homeopathy, but we don't have time to get into that as well.
Well, I learned it wasn't food, it's water, because there's no calories in that. And so the FDA can't quantify it, and therefore they say it's worthless because you can't quantify at the molecular level how things that just make you joyful when you drink water, when you drink food, like those fermented soybeans were disgusting, but they're an aphrodisiac, long life, and the people that eat them every year, so you're cleaning out things and allowing your microbiome to function efficiently. And that's, you know, it's a whole lot easier. And please don't ferment everything yourself. Just use Cardio Miracle. The people that want to do it themselves. I love the doctor who was talking earlier this morning. I realized, and that's integrity. I realized my product didn't do that. So I was delighted. Yes. I mean, I would have loved to have done that at Genius, but Genius didn't. That Anustat only worked in prostate cancer. And now it won't work without Cardio Miracle as a foundation. Now we can do the two, and we've got cures for every prostate cancer. And that's all I really wanted.
Thank you, Dr. Judy Mikovits. You are a international treasure. Love you very much.
Thank you, Robert. Love you very much too.